Hello everyone and welcome to my poster presentation. My name is Andy Folryd and today I will be talking about the GARD skin assay and the testing of chemicals that have traditionally been considered as difficult to test in the current OECD validated assays for hazard assessment of skin sensitizers. So the title of my presentation is Applicability of GARD skin for accurate assessment of challenging substances in the context of skin sensitization testing. So today there is several assays available for the endpoint of skin sensitization, and they have also reached various levels of acceptance by the OECD. But despite this progress, there is certain groups of chemicals that remain challenging to accurately assess in the validated assays. And this includes, for example, pre and pro-haptens, which require abiotic or biotic activations. It also includes hydrophobic substances with low solubility in cell media, and it also includes, for example, surfactants and other substances with severe membrane damaging properties. So the GARD skin assay brings novel elements to the field of regulatory toxicology by evaluating the gene expression changes across a biomarker signatures of genes that are involved in different processes in the skin sensitization pipeline. So following the exposure to a test substance and the measurement of a genomic biomarker signature, a machine learning algorithm is applied in order to classify the unknown substance as either a skin sensitizer or a non-sensitizer. And the assay is now currently under ECVAM review for inclusion into the OECD test guidelines. So the aim of today's presentation is to evaluate the applicability of the GARD skin assay and the potential to actually fill data gaps where the currently validated assays have shown technical limitations, and also to compare this predictive performance across an overlapping data set for both OECD validated assays, but also for some of the reconstructed human epidermis assays. So the method that we used was essentially to select the 12 chemicals that were tested in the OECD validated assays and also in the reconstructed human epidermis. So we selected all the 12 chemicals from a recent publication by Melling, and we retested all these substances in the GARD skin assay. So we evaluated the predictive performance of all the assays against human data, which were available for 10 of the 12 substances. And in addition to that, we were also generating an additional data set of an additional 9 surfactants in order to thoroughly evaluate the applicability domain of the GARD assay for this group of substances. So when we're looking at the results from the testing, one of the most important things to emphasize in the beginning is that when we look at the performance of the OECD validated assays for this limiting data set, we can see that this set of substances were indeed very difficult to test. So for example, looking at the regulatory endorsed 2 out of 3 strategy, which is based on majority voting from the individual OECD validated assays, it was an accuracy of only 40% of this in this data set. So looking at some of the reconstructed human epidermis assays, they were slightly better, reaching an accuracy of approximately 70%. So the GARD skin assay demonstrated an overall quite good applicability for this evaluated set of substances, with an 80% predictive accuracy compared to existing human data. So from the table, we can see that the GARD skin assay correctly identified all pre and pro-haptens and also all substances with low water solubility. So the 2 substances that were classified as false positive was tween 80, which is a surfactant, and also propylparaben, which is actually a human potency class 5 according to Basketer, which may indicate that it's actually have some sensitizing proportions. So the applicability of the GARD skin assay to correctly classify surfactants was also further evaluated on the extended data set here in table 2, and the inability to accurately predict tween 80 from the initial data set was not representative for the group of surfactants, and this was essentially the only misclassified substance across these substances of severe membrane damaging properties. So overall, this conclusion from this poster was that the GARD skin demonstrated an overall high applicability for the evaluated challenging substances with an 80% predictive accuracy compared to existing human data. And the ability to correctly identify prohaptens can be explained by the fact that the cellular model that we use in the GARD assay do actually express several of those metabolizing enzymes required for the biotic conversion of the prohaptens. And the applicability for substances with low water solubility can be explained by the ability of a large panel of non-polar solvents which are compatible with the GARD assay and can also be applied in order to increase the solubility of non-polar substances. So overall, this poster demonstrates that the technical limitations associated with the current OECD validated assay does not necessarily have to apply to the next generation of toxicogenomic-based assays, and that the GARD skin assay may subsequently contribute to fill data gaps and contribute to an expanded applicability domain of non-animal-based testing strategies for skin sensitization hazard assessment. So by that, I would like to thank you all very much for listening to this poster presentation, and I will be happy to take any questions. And also, please feel free to reach out to myself or any of my colleagues at SenzaGen if you have any further questions of this poster. Thank you very much.